Hi there everyone, welcome back to Higher Biology. Today we're going to be continuing on with Unit 1, DNA in the Genome, and we're going to be moving on to Key Area 7, Evolution. And this really comes after our Key Area 6 Mutations lesson we looked at last time, and we looked at the effect of mutation on evolution. We're going to go into more detail on evolution in this key area, and then we'll finish off next time with genomic sequencing. So just to get started, evolution, you'll came across it in National 5. One of our first little descriptions of this that we're going to come across is what evolution actually is. So the statement here says evolution is the changes in organisms over generations as a result of genomic variations. So that all sounds quite complicated in a way, but really, first of all, it's meaning that evolution is the changes in an organism's genetic makeup, but also it's reinforcing this idea that it's over generations. It's not this sort of instant idea that people sometimes think that evolution just happens and all of a sudden you're a new species. It's the changes in organisms over generations because of these differences in their genetics. So what we're going to look at to start off with is natural selection. So you should hopefully remember natural selection, the idea of sort of survival of the fittest by selecting for advantageous genetics or genes from National 5. We're going to go into a bit more detail here, looking at 3 different forms of natural selection. But first of all, natural selection is the non-random, so the chosen increase in the frequency of DNA sequences that increase survival, and the non-random reduction in the frequency of deleterious or harmful sequences. So again, sounds a bit complicated, but what it's meaning is, first of all, if it's a natural selection, something is selected for, it's not this random change in genetics. There's a reason why these sequences have been chosen. So it's a non-random increase in DNA sequences that increase your survival, so that's good genes, and the non-random reduction in the frequency of harmful sequences, so bad genes effectively. It's looking for what genes are advantageous and how do you increase that frequency throughout the species. So as I was saying, we're going to go on to the 3 different types of natural selection. I'm going to give you a brief overview here, and then we're going to go into them in a bit more detail because it can be a bit tricky to get your head right at the start. So we have stabilising selection, where an average phenotype, so your physical description, is selected for. Directional selection, where an extreme of your phenotypic range is selected for. And there's disruptive selection, which 2 or more phenotypes of the extreme ranges are selected for. So that's a bit strange to get your head around just now, so we'll go into a bit more detail, and we'll start off with stabilising selection. So in stabilising selection, an average phenotype is selected for, and the extremes of the phenotype range are selected against. So if we look at this graph just on the right hand side here, if this was the range in an organism's physical appearance, or the physical appearances within a species, for example, what we're looking at here is that at the extremes on the other end, there's a pretty low frequency, but in the middle, right at this sort of average, that's where the majority of these individuals are. When stabilising selection takes place, it keeps that average going. Okay, that's the unchanged average of the phenotype is selected for. So this means that when stabilising selection takes place, there's a smaller range of values, so there's less variation in the population, because having that average phenotype gives that organism a selective advantage. In these videos, because I just try and keep it quite concise, I don't usually go into studies or examples, but for selection I think it'd be quite helpful because it's quite hard to get your head around. So one example of stabilising selection here, we can look at cacti. So if a cactus has a low density of spines on it, if this is what we're looking at just now, if they have a low density of spines, then it's quite common for them to lose their fruits to insect herbivory, so insects eating those fruits that are on the cactus. However, the cacti with high density of spines are often affected by parasites because it gives them a lot of cover and a lot of protection to take over. So if we're looking at the density of spines being a phenotype, one extreme of the range is a low density, the other extreme of the range is a high density of spines, and what we're seeing is both of these have a bit of a disadvantage. You're either going to get eaten by insects or infected by parasites, none of which is overly good. If we look though in the middle, that average phenotype of your density of spines, a regular average density of spines is going to ward off both those pests. And that means that successful cacti, long-living cacti, are in that average range. So being at one extreme or the other extreme of the range with low density or high density is bad. It's not advantageous for you at all. Being in that average is where you're going to survive and you're going to pass on your genes to the next generation. So that would be an example of stabilising selection. The second one we're looking at then, we're going to be using the same sort of graph here, is directional selection. So the best way of trying to get this into your head is that the average phenotype moves in a direction. So in this case, one extreme of the phenotype range is selected for because it now gives a selective advantage. Something has changed, which means that original mean is effectively not as advantageous as it used to be, and a new range, a new part of the range rather, is now an advantage. So there's still a range of values once directional selection has taken place, but the mean has shifted over, it's moved in one direction. So the advantageous trait that was initially quite rare is now more common, because if you have that trait, you're going to survive, you're going to pass on your genetics, which have that advantageous gene attached. So again, another case study for this one, we're going to look at cliff swallows. So during an extremely cold winter, which was a selective pressure through the temperature change, only the large cliff swallows survived because they had a greater fat store. So they were larger, they had a bit more fat, it kept them more warm, they were much more likely to survive than smaller ones. So the larger swallows survive, they pass on their genes for having a larger body, while the smaller cliff swallows, they die, they don't pass on their genes because they didn't have enough insulation going on. So what we're seeing here in this graph is the original population, you had that kind of bell-shaped curve going on, but after this really cold winter, the larger cliff swallows, you can see there's been a bit of a movement here, there's more of these ones. So going back to this original graph here, if this was a phenotype of body size of a cliff swallow, you can see on this side, you'd have larger cliff swallows, this side you'd have smaller ones, and your average would just be your kind of normal, regular sized swallows. But because of the selective pressure of temperature change of a cold winter, all of a sudden what's happened now is that this side, being a bit larger, has been an advantage. So they're going to survive, they're going to pass on their genes. A lot of ones on the other end are going to die off. And now what's happened is that the average phenotype has now shifted to this new average phenotype. So that's an example of directional selection. The final one to look at then is disruptive selection. So you can see from the graph straight away that this is really quite a different one. What happens in disruptive selection is that two or more new phenotypes are selected for. The population is then split into two or more distinct groups, and they have different characteristics. Don't get this confused with speciation. This isn't that they've been split into two groups and they become their own species. It's just a case that within this population of a species, you had your average. Again, something's happened where it's more advantageous to be on either one of the extreme ranges, and then once disruptive selection takes place, you have your new means here where these advantageous traits have been passed on, and the average is much lower or the old average, sorry, is much lower in frequency. So again, we're going to look at an example here, and the one we're going to look at is salmon. So in salmon, if you're a large size salmon, you're better at competing for territories, but if you're a small salmon, you're better at basically fertilizing eggs without being detected, without being attacked or chased off by another salmon. So it's advantageous to be large, but it's also advantageous to be small as well. Where there's not an advantage though is being a medium-sized salmon, because the large have this advantage of being bigger and can compete. The smaller ones are able to be a bit more sneaky, they can fertilize eggs without being chased off and engaging in any sort of competition going on, whereas the regular one doesn't have any of those advantages at all. It's not big enough to compete, it's not small enough to not be detected. So what happens there then is that because both of those ranges give the salmon an advantage, they're going to survive, they're going to pass on their genes. Medium salmon are uncommon because it's not an advantage to be a medium size. So again, what we'd look at here is you can now find lots of small sized salmon and lots of large sized salmon, but it's actually quite uncommon to find an average sized salmon. So what we find here is, just to summarize these different forms of selection, this table is just quite good at remind you of what they are, because what you'd be asked is you could have to describe or to compare the different forms of selection, or given a graph or some extra information, you should be able to say whether that form of selection is stabilising, directional, or disruptive. So that's all you have to know for natural selection. We're going to move on now onto a small part on gene transfer, because I've been talking quite a lot about passing on your genes. So genes can be passed between individuals through two different methods. So what we tend to always talk about, but never really by name, is vertical gene transfer. And this is where genes are transferred from a parent to an offspring, either as a result of sexual or asexual reproduction, and we also refer to this as inheritance. So you came across inheritance before in National 5, and also just through regular conversation. So if you think of vertical gene transfer, it's genes transferring vertically, almost like coming down a family tree. The other one though, which we've not discussed, is horizontal gene transfer. And this is where genes are transferred between individuals of the same population rather than the next generation. Sorry, the same generation rather than the next generation. Prokaryotes and viruses are able to exchange genetic material this way, and there's quite a big advantage to this that we'll come into in a second. So, vertical gene transfer, quite easy to remember. It occurs in eukaryotes, sometimes in prokaryotes as well, and that's where your genes are passed from parents down to the offspring. So, for example, your parents pass on their genes to you through vertical gene transfer, you pass on your genes to your offspring through vertical gene transfer, and so on and so forth. It's important to remember as well, though, that this also happens in asexual reproduction as well. So both sexual and asexual reproduction, whether you have two parents or one parent, if your genes are being passed from a parent to an offspring, then that is vertical gene transfer. It's working its way down the way through the generations, is the way of remembering it. What's different with horizontal, though, is that these members of the same generation can pass genes on to each other. So prokaryotes can inherit genomic sequences horizontally by transferring genes between individuals of the same generation. So they do not have to wait for the next generation of offspring, they can just release it to the same generation they're in. What happens there, though, if you're able to pass on your genes to individuals that are still in the same generation as you, is this can result in evolutionary change and natural selection taking place much faster than organisms that only use vertical transfer. So that's the main takeaway from this part here. You need to know the two differences between vertical and horizontal gene transfer, know a sort of example of them, but the main part is looking at evolution, is that evolution takes place much, much quicker through horizontal gene transfer. You're not having to wait for the next generation, you're just passing them on within the same generation. And that's why you have your bacteria and viruses that seem to just evolve so quickly, because they can undergo horizontal gene transfer. The final part then, I know this is quite a large key area, is speciation. So speciation is something you should hopefully remember from National 5 where we discussed it. As usual, we're going to go into a bit more detail this time of speciation, but hopefully if you don't remember from that 5, it can be useful to brush up as well. So speciation is the generation of a new biological species, and this is through evolution as a result of the processes of isolation, mutation, natural selection, and speciation. So these are all words that we came across before in National 5 and the different parts of speciation that we looked at. One way to try and remember both the key words to use and the order of them is this phrase, I am a new species, where the I is isolation, M is mutation, N is natural selection, and S is speciation. Okay, so hopefully you remember the basics of this. But if you don't, a quick overview here, because it's relatively the same at higher. You'll hopefully remember the idea of an isolation barrier. In National 5 we talked about it quite a lot in terms of a mountain range or a river or something breaking up a population of a species into two subpopulations. So the isolation barrier splits a population. Mutations then take place between both of the subpopulations. Natural selection acts on the new subpopulations as they're normally in a different environmental range now, where there's different selection pressures taking place. And then eventually this goes on for so long that speciation has taken place, and these two subpopulations are now an entirely genetically different group of individuals from each other. So even if that barrier was to be removed, subpopulation A and subpopulation B are totally different species. They could not interbreed with each other. So we're going to look a little bit more though at isolation barriers at higher level. So you'll hopefully remember at least geographical barriers, where we talk a lot about a river or a mountain range splitting up a population. We're going to be looking at some more examples of these and also their impact on forms of speciation that take place. So an isolation barrier of any form prevents gene flow between subpopulations during speciation. So that's what's quite key to remember, is that regardless of what form of barrier is going on, it's stopping gene flow between the two subpopulations. So we have geographical barriers, which we've spoken about before. We have behavioural barriers, and we have ecological barriers. And we're going to go into each example here and their impact on speciation. So a geographical barrier is more of your sort of classical idea of speciation. It's something we discussed quite a lot in National 5. So any sort of feature like a river or a mountain range, the main thing with geographical barriers is that they physically separate a population. There's a physical divide between them. So this is separation of population by natural features, and it results in something called allopatric speciation. So there's two forms of speciation we're going to look at. The first one is allopatric, but we'll come back to that after the barriers, so don't worry about that just now. Just try and remember that geographical barriers result in allopatric speciation. The second one we're going to look at is behavioural barriers. So in behavioural barriers, there's not any sort of physical barrier, physical divide between the two subpopulations. There's a behavioural barrier. So in this case, two populations are capable of interbreeding, but they don't because they have differences in their reproductive behaviour. So for example, here in this picture, you have a western meadowlark and an eastern meadowlark, two different species. Now, what would have happened is at some point, there would have just been a meadowlark species, but they have divided through speciation into the western and the eastern ones. And what's caused that, the barrier that's caused that, is mating calls. So at some stage, a subpopulation would have had a certain mating call and they would all have reproduced together, and the other subpopulation would have had a different mating call and they all reproduced together. Now again, through the same process of speciation, eventually you have two different species. Now, this results in a slightly different form of speciation called Sympatric Speciation. So again, we'll come back to this in a minute, but we've heard about allopatric in the previous example of geographical barriers, and now we're talking about sympatric. So we'll come back to that in a little bit. The final type of barrier is an ecological barrier, and this can be a bit more confusing. This is a separation by an ecological niche. So it could be things like pH levels in plants, it could be salt levels in water, it could be breeding locations being slightly different between populations. And again, this results in sympatric. So we looked at geographic barriers result in allopatric speciation, but behavioural and ecological barriers result in sympatric speciation. So this example here, you have marmots occupy high elevations, but groundhogs occupy low elevations. That it can get a bit confusing and sound almost like a geographical barrier at some forms, but this would be an ecological barrier, some difference in the ecology of these subpopulations led to new species. If you feel like you're getting confused in geographic and ecological barriers, which is understandable, what to think of is in geographic barriers, there's a physical difference. So either a mountain range right through the middle of a population, obviously over time, or a splitting of islands or a river forming, something physically is there. So we've talked about allopatric and sympatric speciation, so let's go into a bit more detail there. What I really want you to take from the geographical, behavioural and ecological barriers is know what they are, know the differences between them, be able to identify them if you were asked what form of barrier is taking place here. But the other thing is that geographical barriers lead to allopatric speciation, whereas in sympatric speciation, the population lives in close proximity, they live in the same environment, but they've become isolated due to behavioural or ecological barriers. So instead of this idea of a barrier such as a mountain range or a river forming between a population, in sympatric speciation, the populations are all living together, but they're still undergoing speciation. So again, we'll go into a bit more detail here. We'll start off with allopatric speciation. Now this is really the exact same as what we looked at National 5. Okay, you have your species, a big population, gets separated by a geographical barrier. That prevents the gene flow between the subpopulations, and then mutations occur randomly in each subpopulation, leading to a new variation in each subpopulation as well. Natural selection acts on each subpopulation because different traits are favoured depending on the selection pressures. So classically what you would talk about is if you had a mountain range as a barrier, on one side of the mountain range it might be quite a warm environment, on the other side of the mountain range with the other subpopulation it might be colder. So straight away there's going to be different selection pressures going on. So for example, in the warmer climate, it'd be more of an advantage to have less fur. You're not going to overheat, you're not going to die off. Whereas the opposite would be true on the other side of the mountain range. If it's cold, it's a selective advantage for you to have lots of fur. So straight away there could be very easy to spot physical differences there that, depending on your traits, your selection pressures will be selected for. Then eventually, as we've said before, the subpopulations become so genetically different through time that they can no longer interbreed. So even if you were just to remove that barrier, speciation's already taken place. Species A cannot breed with species B. That's then where you know there's two different species taking place. Sympatric speciation, though, that we've spoke about already, saying it's a bit different, you can tell just from the diagram here. You have a population of one species occupying the same environment, but this time some members of the population occupy alternative ecological or behavioural niches and they breed. So it's almost like within this one big group, two sort of cliques have been formed. And because of that, two populations exploit different resources, they no longer interbreed with each other, so again there's no passing on of genetic information or gene flow between the two populations, but there's not a physical barrier between them. And really just the same as allopatric speciation. In this case, mutations produce new variation in each group. The new traits caused by mutations are passed on if they give a selective advantage, if they're good genes. And over generations, you then have two genetically distinct species. So you end up exactly the same thing. Two genetically distinct, different species that cannot interbreed with each other, even if they all decide to then start reproducing. And that's it for key area 7 evolution. So I know that's quite a lot to take in, it's quite a long key area and a lot of new information thrown at you, but you need to just know what evolution actually is. Just try and put in your understanding of mutations from last time as well. This result of genetic variation results in evolution. We looked at selection in terms of the three types of natural selection, looked at examples of them. You'd be able to tell them through a graph or some information of what they do, and then comparison between vertical gene transfer and horizontal gene transfer, and that horizontal gene transfer results in a faster rate of evolution. And finally, speciation. We've built on your knowledge of natural 5 speciation. We've looked at a bit more detail at isolation barriers, and then we've looked at differences between allopatric speciation and sympatric speciation. So thanks so much for taking the time to listen to this, folks. I really appreciated all your feedback. If people find this really helpful, it really does mean a lot. I'll be getting on with the final key area of Unit 1 in the next week, because a few people have mentioned that we're coming up to the summer holidays and they're wanting to try and get Unit 1 finished or be revised over holidays. So that's fantastic. Well done you for doing that. As ever, please get in touch with me either here or on Twitter, just if you have any ideas, anything you think would be helpful, and I'll be getting a quiz attached to this as soon as possible as well. So thanks so much, folks, and I hope you're all staying safe. Thank you.